This is the squid dissection component, one of several specimens that we would dissect in class. So I'm going to walk you through a PowerPoint where we do some digital dissection. I'm also providing you with a YouTube link that has a fairly good demo on it where the dissection takes place. Then I have some individual slides that focus on retractor muscles and siphon, the ink sac, the digestive system, the circulatory system, and reproductive systems, male and female. This is the demo. I realize that you can't click on this link, but if you do a search for Biology by Me, Squid Dissection Demo, you will find it. What I did was take clips from the video and illustrate the important aspects. There's an orange arrow here pointing to the retractor muscles. These muscles are elongate, beige, and very easy to identify. It's what you would expect some muscle tissue to look like. Without retractor muscles, the squid would really have to swim by waving its arms and tentacles and fluttering its mantle. It won't be able to expand and contract its body on the long plane. So one thing that the retractor muscles does is it brings the mantle skirt down, pulling it and the buccal mass together. The buccal mass is basically the head. It contains the eyes, the siphon, the mouth. The buccal mass and the mantle are ordinarily detached. That allows for flexibility and constant movement of water over the gills, which we'll talk about later. But what these retractor muscles can do is to make the margins of the buccal mass and the mantle flush. So once the connection is firm, the water collected inside the mantle can be jettisoned down through and out of the siphon, which is in the buccal mass. So that's how a squid can move in spurts through the water. Now I want to talk about the ink sac because it's a very well-known part of the squid. This is the ink sac where it is inside of the dissected squid. This is the buccal mass where the head is. And I just pre-drew this as an ex situ representation of the ink sac. So the ink sac probably originated as an extension of the hindgut. So we regard it as one of those organs that pocketed off from the digestive tract, which is something that only coelomates have the potential to do. And it's an advantage of being a coelomate. It's an added organ that can do something specific and useful. In this case, a smokescreen-like burst of deeply opaque liquid that the squid can move away from in any direction and not be seen during that crucial first moment of its escape. And that, of course, gives it an advantage. The organ is a narrow, silvery sleeve, and it shines in an oily sort of way. When you shift it under the light, it looks different from other organs. It is small, though, and I have seen students have trouble finding it during dissections, but if they really focus, they will find it. The sac has muscles around it, very small muscles, that of course contract during the ink expulsion. So that tightens the walls of the muscles around the ink sac, reducing the volume of the sac, which creates pressure. And then there's a sphincter closer to this end that opens out into the environment. Contract those muscles, relax that sphincter, and the ink will shoot out. So one more bit about this, keep in mind that projection of the ink needs a lot of energy for it to work because an aqueous environment is dense and it's liquid pushing through liquid. That's complicated. The ink itself is very thin and low density, which is better for cloud forming than something that is viscous and oozy. So you should also know that the sphincter feeds into the rectum. So that is where the ink comes out. The ink does not come out the siphon. It comes out of the rectum. So the rectum and the ink sac opening are the same, which is one of the reasons why evolutionary biologists think that maybe the ink sac originated as part of the digestive tract. So it's a narrow passage, which is good for projectile action. The ink itself contains a lot of melanin that has pharmaceutical properties, including antiretroviral and anti-cancer components. So it's studied in several labs in several countries. The ink itself is pretty benign, so it's been used in some food coloring, been used as ink for writing and painting. Ink sacs appear in the fossil record about arguably 330 million years ago because that was the age of the strata where the fossil was identified. So ink sacs have been around maybe for 330 million years, so that places it in the Carboniferous. Moving on, the ink sac is proximal to the retractor muscles that we just went over, and also to the mainline venous system here, which was pre-injected with blue latex. So we'll move on to that now. This is a look at the digestive tract of the squid, and I drew the schematic next to it so you can see some ex situ features. The esophagus is pretty obscured, but you can see it moved when you watch that 22-minute video. Everything gets revealed at some point. But this clip captures the stomach really well. So that's what this orange arrow points to. The author of the video also is poking at this, the cecum. And it's pretty important that I also talk to you about the cecum because, as the videographer pointed out, it's easily destroyed. I've seen a lot of students get confused. Where is the cecum? Isn't it supposed to be this big bag over here? It can degrade and come apart pretty quickly. And when I tell you about what it does, I think it'll make some sense as to why. The orange arrow points to the stomach. The cecum is dorsally located on the squid. Here's what happens. The food item, which is always going to be another animal, whether it was scavenged or hunted, because it's a cephalopod, to my knowledge, all the cephalopods are predators. So this green here is supposed to be kind of fish-shaped. It'll go in through the mouth, enter the esophagus, and get squeezed and massaged up here. The esophageal lining and the muscles around it are flexible. It can eat pretty big chunks. That being said, they don't really swallow things whole. If you look at their bodies, they just about can't. They have hanging, very linear, parallel organs, which means that there's potential for a lot of shifting around to make room for something larger without causing too many problems. However, a squid like this can catch a fish and kill it and start to eat it, and that fish could be half its body mass or more. It does have to bite chunks to eat, but they can be pretty large chunks. So that big chunk can go up this esophagus, which is capable of stretching out a little bit. Then it enters into the stomach. And the stomach pouch, which will vary in size depending on the squid that you're dissecting. It could be very big. In this specimen, it's pretty small. But that is the stomach. It has thicker muscles. That's where digestion starts. And there's some mechanical digestion, breaking apart the food and absorbing nutrients. So then after that's been going on, it continues into the cecum, which is pretty broken up. And it's meant to look fairly thin and sac-like because it is. And the cecum is where most of the nutrient absorption occurs. It can stay in there until it's been milked of its nutrients and then ejected. Or if the squid opportunistically acquires another food item and starts to bring that up the esophagus, it needs to make room. So it may evacuate the cecum contents a little prematurely, but that could be economical because some nutrients have already been absorbed. It's sort of like having a bunch of bananas, grabbing some, eating many bites of it, and rather than trying to eat the little pieces at the bottom, just throw it aside and grab a new one. So the squid has some control over how quickly things move through its system. So that is a bit about the digestive tract. This is the circulatory system, considered to be a closed system. This picture is a freeze frame and an illustration that I did of the venous system, which can seem a little complicated before you understand that the organs are more or less divided into two teams in terms of function. You also have to see margins of the different organs to understand the relationship. So the specimen in the video was single injected with blue latex rather than a double injection where you would see blue and red to represent oxygenated and deoxygenated blood. So I added this. In the video, this is actually blue. I just did this to show you a little bit of contrast. So the gill hearts are also called branchial hearts. They pump deoxygenated, that's why they're blue, deoxygenated blood down into the gills. There is a mixture here. I have blue and I have red. because there is deoxygenated blood coming down. There is also oxygenated blood being forced up, largely by the pressure. This is a closed loop. If there's downward pumping constantly, some stuff is going to get pumped back up. So that's what's happening, and that's why there's mixture there. So the gill heart, also called branchial hearts, they pump the deoxygenated blood down to the gills, and the gills come into contact with the ocean and the oxygen in it. There are little tabs called lamellae organized in these gills. That's where the oxygen is absorbed. The lamellae are packed with vessels that are right at the surface. The lamellae rows are aligned by a main vessel, and the whole thing is contained in a leaflet-like packet. It's a bit of a membrane. The mantle is pumping, and that's what keeps new water flowing up into the cavity and over the gills. So now back to the gill hearts for a second. They pump deoxygenated blood down, and as I explained before, that forces some back up. And where it goes is into here. This is the systemic heart. And there's some faint margins that are difficult to see in a video, but I tried to illustrate them here. Here's one line, here's another partition. And so you really get 1, 2, and 3 pieces. So the systemic heart does what our hearts do as well, which is to pump oxygenated blood to where it is needed in the body, including the retractor muscles, around the digestive tract, to the reproductive system. This is delivering the needed oxygenated blood. So after it's been in the body, it'll start to collect in these post-vena cava. There's one on each side, and that will feed it back in. That is how it gets back into the gill heart, and then it pumps back into the gills, and it starts all over again. So to recap a little, the systemic heart delivers blood to the muscles and everywhere else it needs to be. The blood will cycle back to the posterior vena cava, where it is then delivered down to the gill heart and down to the gills, up the gills, to the systemic part, out in the body where it is needed, to the vena cava, down to the gill heart, and on and on and on it goes. This is a very simple piece of anatomy. The pen. I found on the beach sometimes. It's like a piece of plastic. The videographer is pointing to it with the probe, and it is this brown piece right here, and it actually runs the length of the squid's mantle. So it's long and plastic-like. It can be brown or purplish. It helps maintain some rigidity. Octopuses, also cephalopods, they don't have pens, and that's part of the reason why they can squeeze through extremely small openings. Cuttlefishes, another cephalopod, they have a calciferous, buoyant cuttle bone instead, chalk-like, and we give them the pet birds, primarily parrots, because they instinctively scrape their beaks on it and it keeps their beaks healthy. Another cephalopod that we pass around in lab that's very cool to look at, we have their shells. It's a flexible squid-type body in a heavily partitioned shell. I'm talking about the chambered nautilus. Like all cephalopods, it has a siphon, and it descends the water column by letting seawater into its chambers. It ascends by emptying them. So it moves horizontally with some retractor muscles that control the siphon's aim. So they're predators of anything smaller than they can catch. And they're food for predators that can break through the shell. The nautilus does not have a pen. The octopus does not have a pen. The cuttlefish does not have a pen. Squid has a pen. This is the reproductive anatomy of a male squid. You'll recognize some of the names here as we use them on humans. Here's a testis, singular. There's only one. This is the vas deferens, which the sperm must pass through on the way out of the body through the penis, which is obscured here, but you can see in the full video. So the testis tends to be dense and yellow and quite easy to identify. Fertilization is external, and the gametes join together beneath the mantle of the female squid. They grasp each other with their arms and tentacles. So the penis has rigidity and can be protruded and controlled by the male squid. So in terms of its origins, evolutionarily, the squid penis is theorized to be an elongated, well-developed gonoduct. So the whole thing was delicate and internal at one point. But for squids, and some species have a very short penis, some species have a long penis. It depends on their lifestyles. At one point, the penis was delicate and internal, but now it's useful for this reason. Again, has to do with being in an aquatic environment. It has its challenges, and having a penis helps overcome those challenges if you are choosing your mate. In other words, you're not spawning. So it's useful because the sperm cells themselves, which are quite vulnerable, lightweight, and they can't do much to move independently besides wave a flagellum. They aren't released on their own in the squid. Instead, they're rolled into bundles called spermatophores on the way out of the gonaduct. So the sperm aren't released in a cloud of semen that contain a non-unified horde of diploid male gametes, but rather the squid releases something more like sperm cannonballs. So those are the spermatophores. The penis is not very visible here, but watch the video, it is shown. And the vas deferens, again, is where the sperm travels to from the testis before it is released into the environment. That is the male, and this is a female. And I had to go to another source to get it. You could get a question where you need to tell whether a squid is a male or female. And depending on the specimens, that can be extremely easy or it can be a little bit challenging. So I'll talk to you about it. So this is a female squid, and you can see what the ovary, when eggs are well developed, looks like right here. So if you opened up the squid and you saw some honeycomb looking stuff right up here that's shiny, it may look a little like roe, R-O-E, fish eggs that we eat, then okay, that's easy, that's a female. So then it's easy to identify. There is one ovary, just like there is one testis. At another time in the squid's reproductive cycle, this honeycomb may look solid and white and a little bit like this. It isn't always this obvious. The testis, though, isn't usually as high up, not as dorsal, and it is not as pointed at the top, at the apex. There's also a lack of vas deferens. There's no bulgy vas deferens. And I've saved the best part for last. I've made it sound really hard, but it gets a lot easier when you realize, oh, okay, presence of nidamental gland. That means it's a female. It's very conspicuous. Males don't have a nidamental gland because they don't need one. The nidamental gland is a female reproductive organ. It's considered an accessory gland, which is an organ that contributes to the function of another organ. In this case, the nidamental gland is involved in egg case secretion. It enables the eggs from the ovary to survive in the environment. That was a summary of the squid dissection. I hope that you watched the full video, and it can't hurt to do a few illustrations of your own. It really helps understand what it is you are looking at and how it all fits together.